So hello, everybody. Are we good to go? Yeah, okay. So thank you, everybody, for coming here. It's a great pleasure to receive Dr. Leandro Araújo. Dr. Leandro is the recently hired professor here from the Department of Immunology of the Institute of Biomedical Sciences at the University of São Paulo. Dr. Leandro Araújo is a biologist who had his PhD concluded in microbiology and immunology at Federal University of São Paulo. Then he left for a postdoc at Columbia University, also at the Department of Immunology and Microbiology. And now he's back here and he's going to present his data. His talk is entitled Dissecting the Role of Type 3 Immunity in the Mucosal Protection. So, Leandro.
So thank you, Diego. Everybody can hear me. I have to talk higher than I was praying. Basically, I'm very happy to be here to know everybody. I have met a lot of people in the corridors, but I didn't have the opportunity to talk with you guys before. I'd like to thank the organizing, Gustavo and Diego, for the invitation to talk a little bit about my work I did in the postdoc, and I talk a little bit some data from my PhD too. And the plan is try to connect everything I did during my postdoc and PhD and show you guys what I plan to do here in the Department of Immunology. So I start showing, first of all, I was very interested since my PhD about the interactions of bacteria and the host interactions. So I previously worked with the EA model, so I have helped some students from university from USP, and I always was curious about this microbiota interaction with the host. So that's why I plan to go to the Columbia University, and I did my postdoc in the Immunology lab. So the first piece of data I will show you guys these interactions with the bacteria interactions with the with the host. So the title, as you will say, Dissecting the Role of Type 3 Immunity in the Mucosal Protection. So basically, I work with Th17 cells in the ILC3s during this period. And the first piece of data I show you how the specific microbes or commensals can interact with the host. Okay.
Yes, perhaps.
I can.
Yeah, good with.
I can do it.
I think it just.
But try again. So now it's working. Sorry. So the microbiota shapes the immunity. So we know that working with the mucosal, especially the intestines. So here I'm showing this model. So we know that the pathogens and the hosts have this difficult challenge to distinguish pathogens from commensals. And plus we have the diet. So the intestinal mucosa have to absorb nutrients from the diet too. And basically have the immunity has to kind of distinguish these two bugs, pathogens from microbiota, so beneficial and pathogenic bacteria. So ILC3s, they express RO gamma. They can, one of the roles of them, they can delete specific T cells that are against the microbiota. So this is one of the main mechanisms they can interact. They can produce IL-22 and inhibit the pathogens in these intestines. And we know that the great cells can produce IL-10 in this context too, and kind of keep the tolerogenic environment to keep the microbiota and absorb nutrients. But at the same time, we have showing the literature, we have specific bacteria that can induce different types of T helper cells. So Clostridium members can induce specifically regulatory T cells, and these regulatory T cells can control releasing TGF-beta and IL-10, the other cells against pathogens or against commensals. So we have bacterial fragilis that can induce specifically Th1 or Listeria. And specifically here, I want to show you guys a specific role of how the SFB segmental filamentous bacteria can interact and induce Th17 cells. So in humans, we have a lot of piece of data showing that specifically for this induction is very important, the epithelium-associated microbes. So we have SFB and other works specifically from this group that is very important that work with microbes, Kenya Honda's lab. They demonstrate that, for example, in humans, they have like 20, combination of 20 strains that could induce Th17 cells. And the main thing these bugs, they have to interact or attach to this epithelium to have this Th17 cells induction. So as immunologists, we every time look for the host microbe interactions, and we know a lot of how these interactions could happen. So we could have like receptor engagement. So for example, toll-like receptors or not-like receptors could recognize bacteria. We have like some bacteria that could, using type secretion system, evade this system. So in the case of Salmonella and Shigella, we could have the internalization of these bugs from the phagosomes, and specifically some toxins like could be endocytosis by mediated by clathrin or caveolin or other mechanisms. So the main question here in the first part, we would like to understand how the commensals could interact with the host. So we know a lot of how the pathogens can interact about specific for using receptors or internalized by using other receptors internalized by phagosomes and using type secretion system and endocytosis, but we know very little about how commensal microbes could interact with the host. So here we use as a model as segmental filamentous bacteria is a bacteria that lives in a lot of different animals like mice, or they don't have like human counterparts yet, but they have they basically can induce intraepithelial lymphocytes. They can induce IL-3 to produce IL-17, IL-22. They can induce Th17 cells. They could induce plasma cells to produce IgA. So to perform this analysis, we take advantage of the electron microscopy. As you can observe, we appreciate here in this figure, we have SFB here attached to the intestinal epithelial cells. And basically analyzing these structures, we can see here in green SFB, in purple we have intestinal epithelial cells plasma membrane in purple. So if you cut here, we can analyze this hold fast, is like a hold fast interaction. This SFB kind of penetrates inside intestinal epithelial cells. And as you can appreciate here in this movie, this reconstruction of this interaction, we have these blebs released inside the intestinal epithelial cells, especially the tip of this interaction. When we did this reconstruction, we can observe like, looks like some kind of vesicles released inside the intestinal epithelial cells. So we'd like to understand next how these vesicles, characterize better how these vesicles are. So basically, as you can observe here, we have SFB and we have these vesicles, this hold-fast tip. And basically analyze the size, the shape, and the origin of these vesicles. We could conclude that it is like an endocytosis process occurring here. So here we have BAC6 and NSG mice. So this happens independent of the presence of immune system or not. So basically, because it's a new microbe interaction, we name this process like microbial adhesion triggered endocytosis or MATE. And we would like to understand how this happened more specifically. So after we could observe in higher magnifications, we can see like a cargo particles inside these vesicles. So here we have SFB in purple, in green we have intestinal epithelial cells plasma membrane, and inside you have like cargo that looks like from SFB origin. So we next ask what is the nature of the main cargo, and do the vesicles contain cargo with bacterial origin? So to see specifically how the SFB could be localized inside this, we use antibody specific to a cell wall associated protein that is called P3340. And we use immunostaining with gold particles. So we can see here like these proteins immunolabeling inside the cells. We can see here like these vesicles looks like in the apical part, in the basal membrane, and the basolateral. So it's kind of spread out around all the inside the intestinal epithelial cells. And we can see that there is inside these vesicles here. So the next step, we would like to understand what are the mechanisms that is involved in this vesicle formation. So I didn't show here, but we saw that there is a Cathepin independent pathway. So we start to look in the Cathepin independent pathways to look what are the proteins involved in this process. So basically one important candidate was CDC42, is control domain C42, is a GTPase that are important for the vesicle formation too. And to kind of demonstrate how this happens, we did a assay that is called intestinal loop assay. So basically we expose the intestinal loop, and inside this lumen we treat this with specific inhibitors for the pit stop inhibitor for patrin inhibition, KZ1 that is for CDC42, and dinosaur is specifically for dynamine. So after analyzing the tomograms here, we can see that there is this disturbance of this vesicle formation when we use the CDC inhibitor, and we have the same thing with dinosaur. In the Cathepin inhibitor, we didn't have any perturbation. When we analyze these vesicles, they have huge numbers showing that this kind of perturbed by these inhibitors. So to test if it's CDC42, specifically this pathway, we generate a conditional knockout mice. It's called CDC42. Basically using CDC42 flox mice, crossing with Villain-Cree mice. And when we did that and colonized these mice with SFB, we couldn't see this vesicle formation like the wild type. And when we stain for the P3340s, the SFB antigen, there is very decrease of these SFB proteins inside these vesicles. So the next question, we'd like to understand how is the Th17 cells induction this process. So we did analyze the IL-17 expression and the induction by the FACS staining. So as you can appreciate here, we have a huge decrease in the Th17 cell induction in the mice that don't have CDC42 in the intestinal epithelial cells. And when we did transfer SFB specific transgenic CD4 T cells for mice that are colonized with SFB wild type and CDC42 knockout mice, we analyze after 4 days the proliferation. As you can see here in blue, we have decreased T cell proliferation, SFB specific T cell proliferation, in the CDC42 conditional knockout mice. So basically here in this work, we demonstrate that SFB uses adhesion trigger endocytosis or made to transfer this antigen inside or through the intestinal epithelial cells. This mechanism is dynamine dependent and CDC42 dependent, and these vesicles travels inside the intestinal epithelial cells, and then can induce Th17 cells. So when we block CDC42 in this context, we didn't have the formation of these vesicles, and we have a small induction of Th17 cells. So the next we'd like to understand or dissect specifically how the T cells and ILC3 contributions in the gut immunity specifically, because as you can appreciate here, we have different type 1 and type 2 and type 3 immunity. We have for each Th17 cells, T helper cells, we have the counterparts in the innate lymphoid lineage. So we have Th1 that express Tbet and produce interferon gamma that are important for the viruses, bacteria, and immunity response. We have Th2 and ILC2s that express GATA3 and produce IL-5 and IL-3 that are important for helminths and allergy. And specifically in this work I'm gonna present now, we have ILC3s that express RO gamma and Th17 cells. So basically the innate lymphoid cells, they don't have specific T cell receptor or B cell receptors. They express partner recognition receptors. They can recognize bacteria by these receptors, but at the same time they behave like a effector cell, producing IL-17 and IL-22. So here's just how these cells, because they are kind of new, and how they developed in the presence of specifically the IL-7 cytokine induction. We have different subtypes of ILC3s. We have a specific one that is called lympho-tissue inducers. They can be important. They are important for the lymphoid tissues, secondary lymphoid organs formation. They are dependent of lymphotoxin alpha and beta. So without this population, we don't have the formation of lymph nodes and Pires patch. And basically, all these cell types, subtypes, they express RO gamma, but they express different other complementary transcription factors that could differentiate in different types. So in the HR and RO gamma, we have the population that is called NECR negative or ILC3s that don't have any KP46 expression. And when you have Tbet expressed in this during this development, we have the NECR positive ILC3s. Basically, all these subtypes, they respond to IL-1 beta and IL-23. They produce IL-32 and some produce IL-17 too. So to characterize these cells are very difficult because they don't have a lineage marker. So we have to stain for different lineage markers and put in dent channels to analyze these cells. So we have here, as you can appreciate here, we have lineage 1 and lineage 2. So we exclude B cells, T cells, myeloid cells, dendritic cells. And in this lineage gate, negative gate, we can analyze the whole ILC3 populations, the total ones, looking for the expression of IL-7 receptor and CD90.2. And when we look specifically for the transcription factors, like Tbet, the ILC1 express, Tbet's here, so we can see here. And we analyze GATA3 and RO gamma, we can distinguish between ILC2s and ILC3s. And as I mentioned before, we can subdivide the ILC3s in at least three subtypes. There is NKP46 positive, one is NCR positive, and the CCR6 positive cells. So basically, they develop very early in the development. They help to the formation of the lymphoid tissues. They can reside in lymphoid tissues too, but mainly the ILC3s, they are present in the mucosal, in the intestinal mucosal tissue, in the Pires patch, and in the large intestine, we have this cryptopatch that is like inside lymphoid organs, inside the lamina propria, that they are present too. So they have this being described with different functions in the literature. So we can have, for example, with the metabolites or circadian rhythm, they can produce IL-32 and can be important for the homeostasis or homing these cells in the lymphoid tissues. They can produce IL-22 and IL-17 in the presence of inflammatory cytokines. They are very important for the induction of antimicrobial peptides and for the barrier in function. And with the IL-17 production, they can be important for the neutrophil recruitment too. And more recently, they have been demonstrated to produce IL-2 and helps the function and induction of Tregs for the immunoregulation in the gut, and for the GM-CSF production and keep the myeloid homeostasis. So basically, as I mentioned before, these cells are very difficult to distinguish the functions of from T cells. And one caveat of the field is we don't have a specific ILC3 knockout mice. So basically, most of this data that I showed you before, they are generating RAG knockout mice. We call the like a disparate chimera. When you reconstitute these RAG mice with Ty1.1 T cells or B cells, we treat these mice with anti-Ty1.2 depleting antibodies. So basically deplete not only ILC3s, but all the ILCs, as I show you the guys before the flow cytometry, how they express Ty1.2. And basically they have like T cells and B cells, but they don't have ILC3 and all the other ILC, innate lymphoid cells. So the main questions here we'd like to answer is what are the specific functions of ILC3s in the gut immunity? How the ILC3s function in the presence of T cells, and are the T cell responses similar in the absence of ILC3? To perform these experiments, we generate a genetic specific mice that we call RO-gamma stop mice. We invert the RO-gamma expression gene, and we flank this or lock this expression with two lock sites. One is normal or regular LOX-P, and one that we call stop. So basically, we can basically in these RO-gamma stop mice, basically they behave like RO-gamma knockout mice, like a full knockout. So when we cross these mice with CD4-3, we turn on the RO-gamma expression in the CD4 T cells, but they still are off in the IC3s. And basically the T cells, we put this gene in the correct orientation. So that's why we have the RO-gamma expression in the T cells. So the first experiment we did is very important for the T cell development of these cells, because we do like to understand that these mice, we have normal T cell development. And at the same time, we want to show that this is really a knockout for LC3. So this is the stop RO-gamma stop mice. They behave like RO-gamma knockout. They have a defect in the single positive CD4 T cells, and they have a defect in the frequency of the maturation of double positive lymphocytes. So when we cross the CD4 mice, we recover this CD4 positive single cells, and the double positives too, as you can see in the graph. So next, we want to understand specifically in the gut, how is the Th17 cells induction in the ILC3 absence in these mice. So here is like in the T cells, we get the CD4 positive and TCR beta T cells. And as you can appreciate, we have the RO-gamma expression of FOXP3 expression. In the stop mice, we have the loss of RO-gamma expression T cells. But in the CD4 free mice, we have the recovery of the RO-gamma expression CD4 T cells. So next, we want to understand the ILC3 absence in this model to confirm if it's really ILC3 knockout mice. So here we have the ILC3 gated in the lineage negative analysis GARA3 in RO-gamma expression. So here we have the stop mice don't have ILC3, and even the stop CD4 mice. So basically showing here that we have mouse specific knockout for ILC3. So the T cell development is recovered in all delta ILC3 mice. So we call these mice delta ILC3 mice. Th17 cell differentiation is restored in the delta ILC3 mice. ILC3 are absent in the delta ILC3. But one big problem we have here, the mice don't have lymph nodes in pyropatches, and I mentioned before, because they are whole ILC3 knockout mice. So we have knockout here to the lymphoid tissue inducer cells. And I will show you guys how we overcome this problem later. So basically, this is just the RNA-seq analysis to show that we recover all the cytokines and receptors and transcription factors in Th17 cells. In the intestinal epithelial cell, that is very important. We show here that really ILC3s control the antimicrobial peptide production in the presence of T cells. So here we can see here that we have the effects in hereditary gamma FoT3, SAA2, and other antimicrobial peptides. So basically, we have a mice here that is the CD4 and the wild type are very close in the CD4 T cells, and the mice that is absent in the ILC3, they are very similar to each other. So the conclusions in this part, we have ILC3 control genes are down in stop and delta ILC3, so the mice that don't have ILC3s. IL-22 response genes are down regulating ICs and off stop in delta ILC3. And basically, Th17 cells programming is restored in the CD4 T cells. So next, we want to understand in the Citrobacter rhodesium, that is a classical model for this study of ILC3s and the gut immunity. So in the literature, what we have is specifically the ILC3 are very important in the beginning of this infection. So they produce IL-22 in the beginning, and after a few days later, the T cells took over of this process and produce IL-22 and clear these pathogens after at least 2 or 3 weeks. So when we analyze the Citrobacter rhodesium infection, this model, as you can observe here, we use a high dose of Citrobacter. The mice that don't have ILC3 all die between around day 8. But when we do like a low dose, we can observe that the CD4 mice, they could survive longer compared to the delta. The delta ILC3 mice survive longer compared to the stop mice, showing that the CD4 T cells have some role in this beginning too. So, but the problem here, all the mice, they don't have lymph nodes, and the wild type mice, they have lymph nodes. That's why we saw this difference here, that wild type surviving during this infection, and these two groups that don't have ILC3s, they die. So to kind of put the mice in the same conditions, we use like a raw gamma knockout mice, and we did like a bone marrow chimera in these mice, transferred these different bone marrows from the wild type, stop, and delta ILC3 mice. So basically, all the mice don't have lymph nodes. The wild type have the ILC3s, and Th17 cells, stop, don't have both cells. And the delta ILC3 mice just don't have ILC3s, but they don't have lymph nodes too. Put in this condition, we fed these mice with Citrobacter rhodesium, these chimeras. So as you can observe here, we have difference. We can still have the difference between stop and delta ILC3 mice, showing that T cells are important, but the lymph nodes are very important too for this infection, showing that we have like a overlap, lymph nodes and T cells are really important for this infection too. The next, what we did is just to show that it's like IL-22 specifically. So these differences, we treated the depletion IL-22 cytokine, and we lost this difference between the delta ILC3 and the stop mice. But even this is the chimera set too. So even with IL-22, the wild type mice die too without lymph nodes. So the conclusions of this part, ILC3 are required for protection at high dose. Both ILC3 and T cells contribute to early stages of low dose infection, and lymph nodes are required for clearance of Citrobacter rhodesium infection. So to overcome this problem, we generate another I'll try to explain, very complicated strategy, but to work it. So I cross the Arogama flox mice with a CAR-3 mice. This is a ubiquitinous screen that is tamoxifen inducible. And with these mice developed the lymph nodes during the development, and after they develop the lymph nodes, I treat with tamoxifen. So basically, I deplete gamma T cells and ILC3s, and to confirm that they don't have both, I irradiate these mice at 8 weeks old, and did a bone marrow chimera with these different bone marrows, wild type, stop, and delta ILC3. So basically, now we have like a model that all the mice have lymph nodes, but they have different contribution of ILC3s and K17 cells. So, but first we just confirm that these mice restore the K17 cells for ROGAM expression T cells, as I showed you before. They don't have ILC3s, both stop mice and the delta ILC3 mice. And when we start doing the infections model again to test the role of ILC3s, the differential contribution of ILC3s and K17 cells, we did a high dose first. The mice still die, showing that ILC3 is really important for the beginning. And as you can appreciate here, we have like, basically this is IL-22 dependent because the mice don't have ILC3 and the IL-22 production in the ILC3s. But when we did like a low dose of Citrobacter rhodation, so basically these mice survive. So showing that the ILC3 are important, but it's dependent of the context of the dose and the contribution of T cells and the ILC3s. The mice, they kind of suffer a little longer, showing that the ILC3 is important in the beginning too, even in the low doses, but they can kind of catch up in the end that they clear the pathogen. So this is just to show in the T cell how it works. So we have a defect in the absence of ILC3 of the Th17 cells and IL-22 production. But when we have like in the later stage in the after 14 days of the infection, we have the cells start to produce IL-22 and IL-17 normally, and they clear the pathogen. So the conclusions of this part, we generate ILC3 specific deletion mice. ILC3s are required for protection of Citrobacter rhodation at a higher doses. J17 cells can provide protection for Citrobacter rhodation infection at low dose. And we can study the relative contribution of ILC3 in T cells in the mucosal immunity. So I didn't have time, but I'd like to understand, especially the secondary response, how this happens, because I have one hypothesis that ILC3s are important for the secondary responses for vaccines or in the like intestinal mucosa. So just to complicate a little more, I just show a few pieces of data of my PhD, so I can talk what I did before and what I kind of plan to do here in the Department of Immunology. So in the PhD, under the supervision of Alexandre Basso at the University, we did like we'd like to understand how the sympathetic nervous system can modulate like immune responses. So we know that the nervous system can interact with the immune system at least for 3 axes, like the vagus nerve, the sympathetic nervous system, and the HPA axis, producing corticosteroids. We know that sympathetic nervous system can produce catecholamines, and basically the immune cells can express adrenergic receptors. So we have the most expressed catecholamine adrenergic receptors are the alpha-2 and the beta-2 adrenergic receptors. So here we have a noradrenaline. So we have like macrophage dendritic cells can express adrenergic receptors, B cells and T cells. They are G protein receptors. They have the subunits, different subunits, depend on the type of the receptor we have, like inhibitory, activatory, G stimulatory function. And we basically would like to understand the experimental autoimmune encephalomyelitis, how these interactions can happen. So EA is like animal model for multiple sclerosis. They are Th1 and Th17 cells can induce this disease, and they are specific for the central nervous system. In this work, what we use as a model to study sympathetic nervous system role is the hyperactivity of the sympathetic nervous system. To do that, we use a mice that is called adrenergic receptor alpha 2 NC knockout mice. It's a double knockout mice. Basically, the alpha adrenergic receptor, they are important. They are expressed in the presynaptic fibers. They can signal the norepinephrine and stop the release on new norepinephrine. So basically, knocking out this receptor, we have the increased production of norepinephrine. And we do like to understand the role of this hyperactivity in the EA model. So basically, as you can see here, we have a decrease of sympathy in the EA induction in these mice. When we analyze TH1 cells, we can observe here like less production of interferon gamma and TH1 frequency of cells. When we transfer T cells for this environment, this hyperactive environment, these cells can decrease this proliferation rate in the presence of the sympathetic high sympathetic nervous system, as you can observe here. And basically, what we demonstrate in the work that in the adrenergic beta 2 receptor is very important for this role of the sympathetic nervous system. So basically, transfer specific MOG encephalotogenic T cells that are knockout for beta 2 adrenergic receptors. We have lost this phenotype of the modulation of EAE in the presence of sympathetic nervous system. So this is just the model that we, the graphical observed showing this data, summarizing this data. We have like hyperactivity of the sympathetic nervous system with the noradrenaline release increased. We have the similarly via beta 2 adrenergic receptor. So basically, we have here less interferon gamma, GM-CSF, and IL-2 production, and small inflammation in the nervous system. So basically, what I plan to do here in the department, I try to combine these two areas that I work in the PhD and the postdoc. So I want to work with the gut-brain axis, and basically, one of the ways I kind of, this conversation is bidirectional, so I'd like to understand both ways that it could happen. So one way I would like to understand how specific microbes like SFP, or when I modulate specific microbes with high sugar diet, we have a work published in my old group that we show that we have different bacteria those, when we treat the mice with high sugar diet. So modulating these different microbiota or specific communities, how this could cause an inflammatory neuroinflammation in the central nervous system. So this is one way I'd like to understand. And basically here, I think the innately lymphoid cells are kind of the sensor and the bridge between these two systems. So basically, they express adrenergic receptors, they can respond to microbes. So I think they could be a very important cell type that could do this conversation, one of the ways they could talk. The other way is like when the brain could controls like during the, for example, inflammation, infection like Staphylococcus aureus infection. So IC3s, they express some of adrenergic receptors, and basically the sympathetic nervous system can modulate this immune response via ILC3s and J17 cells during this process. So this is just one preliminary data that I have from this part that I did when I still was in the US. One very specific model we have, I didn't mention before, sorry, for the ILC3s, where is the deletion of MHC class II in these cells, because they express aerogramma and MHC class 2 are expressed mostly in the innate counterparts. So and when we cross the mice, MHC class 2 flux mice with RO gamma GT3, we could delete this MHC expression in the ILC3s. And what I did, I just induced AA in these mice. As you can appreciate here, we could observe that in the absence of MHC class 2 in the ILC3s, they cannot induce a proper EAE response. So my hypothesis here, they are very important sensing these microbes that are important for the EAE induction too, and they probably could even besides that they can present encephalotogenic antigens to the T cells, and we have like less EAE induction. So the other part I would like to work is basically Jimmy Talbot is a former postdoc from Dan Leitman. He's a Brazilian from Ribeirão Preto. He did demonstrate that ILC3s, they express adrenergic receptors like alpha-1 beta and alpha-2A. So this is one thing I would like to understand is during the Citrobacter rhodesian infection, how these receptors are important for this model in the ILC3. So first, I will do in the total knockout mice. So I have these mice from the Basel. Will kindly provide these mice for me to do these experiments. But I plan to buy like a flux conditional knockout mice and cross these mice with GT3 to specifically delete the actual receptors in the ILC3. So I'm happy to take any questions, and I would like to ask students if they are interested in these areas. I mentioned before what I work in my line of research. They can contact me and we can talk about the possibility to do like master PhD or scientific initiation. So I'd like to thanks for the attention.
First, why is Denise started? I don't know if there are students from Rio and Berlin.
No, I have a question. Do you know which type of IX that SFP invades, or it's any type?
Sorry, say again.
Which type of intestinal epithelial cell that SFP invades, or is any type?
We did it. Basically, they are very specific for the terminal ileum, but I think they don't have. I don't know if they have a specific epithelial cells that they could interact, but they mostly are from the terminal ileum epithelial cells.
And what is the reason? Because it's mostly for the terminal ileum.
I don't know. Probably it's like evolutionary or something like that. They could. We have like we kind of asked some questions about this before. We kind of know that. I think they depend on some probably nutrients or something from the intestinal epithelial cells, and I think probably the terminal ileums we don't have like digestion and something. So I think that's why they probably reside there, but probably it's like something that we don't know now that they kind of evolutionarily they localize this specific area of the gut.
And another question, you have shown that this is important to mice for the induction of protective Th17 cells for barrier reinforcement, for instance. What is the mechanism similar that happens in humans?
I don't know. So I have a previous friend in the Ivanov lab that she worked with like these cells, try to show you the mechanism of this. So basically what she saw is these cells can produce, besides IL-17, they can produce IL-10. So probably it's one of the mechanisms, but right now we don't know any kind of bug that looks like SFP that is present in humans. So the ones that some people kind of think is like bifidobacter, but they don't they kind of look similar, but they are completely different. So I don't know if you still have, but my hypothesis could be like IL-10 that could be protected for especially for the Citrobacter elevation. Is very well demonstrated that if you have SFP, I didn't show this data here, but I have. If you have SFP in the mice, they are more protective against another infections, like Citrobacter infections.
So Miriam is saying there's a question from Marcelo Boza. You wanna, actually, Bruno is saying that, you wanna open up the mic?
Hi, Leandro. Great talk. So, yeah, I have two short questions. One is related to the microbiome in the Delta ILC3 mice. I mean, you have shown clearly the role on Citrobacter rodention, but your original question would be, what is the role of LC3 on the microbiome? And have you looked to that? And the other one would be what the vesicles that are profoundly affect when you block CD42 has. I mean, what are the content of these vesicles in the SFB interaction with epithelial cells?
Thanks, Marcelo, for a nice question. So in the first question, we did analyze some the microbiota composition. Basically, what they have, specifically SFB, they have higher numbers because SFB, the ILC3 is very important for controlling SFB. And we have another work from the Japanese postdoc in the Ivanov lab that he showed like other bacteria could grow in these conditions to or even in the high diet. I didn't look carefully, like not carefully. I have this data, but I cannot mention here. But they have different types of conventional distribution. So, and for the other question, sorry, say again. The last is the CDC42, right? Yeah, I don't know. Probably the composition. I don't know for sure. We know that these vesicles, they go for the phagocytosis pathway too, because we have like phagolysosomes in the basolateral or the basal membrane. One thing we didn't yet, we know probably will be published soon is that what are the dendritic or macrophage contribution for this process. We didn't show in this paper because it was very complicated at the time. And we have like different knockouts for use like ZBT46, like for dendritic cells, for macrophages like CCR, DTR mice. And what looks like that the dendritic cells are important for this acquisition after they go to the lymph nodes and induce Th17 cells. And to keep the Th17 cells in the gut, the induction, we need the macrophage too. So basically, we need both cells to keep this induction. And I don't know when, but very soon, you guys probably will see which kind of dendritic cells is present. I cannot say now because we are still finishing some work about that, but we know some of these pathways. Thanks for the question.
So any other questions, guys?
Actually, I do have a question in the C-rod infection model. I was kind of expecting that the ILC3 deficient mice would die faster than the CD4 based on the kinetics that you first explained, but they die later. What do you think is going on there, and why there is after that long the Th17 cells do not take over and protect these mice?
Yeah, so basically what I think is happening, basically what we show in this work, the ILC3s are still important for this protection, but looks like probably we have like redundant pathways, like probably like dendritic cells or other antigen presenting cells could take over from this beginning without ILC3s, but they don't have enough time to prime or to induce a Th17 cells response. That's why the mice die. But probably when you have low doses, we still have time to kind of the immune system generate T cell response. But even in the absence of ILC3, that's one thing I for sure is not I demonstrate, but I think the ILC3s are really important for these Th17 cells inductions too, because as I show you guys before, we have a kind of delay, right? Especially for the IL-22 production. That is the main cytokine that is important for this model. So I think somehow the T cells could receive these antigens by other antigen presenting cells in the absence of ILC3s. That's why they could catch up and kind of clear this pathogen after a few days. But it's kind of delay, right? So the mice, I didn't show here, but they have like higher inflammation during this period. They kind of lost weight. So they kind of suffer. So probably IL-3 is really important too for not just for the beginning, but for the later response to Citrobacter relation.
Right, thank you.
Okay, so we have two questions here, one from Vinicius and one from Montiel. And then easy to. So.
Thank you very much.
Denise first, Vinicius. Montiel.
About the immunodeficient deficient ILC3s and also in their full knockout mouse. So your data on the EAE, do you think the ILC3s could be working as an antigen cells to promote occurrence, similar to those new studies on the organ T expressing?
Yeah, that's
could also be another subset of Treg.
Yeah. So that's a really important thing that could happen. So I think is the rodents group, they show like the expression of R gamma in the cells are really important for specifically for the inflammatory cells. Yeah, I think probably for sure for commensals, they do that. They present antigens, but we didn't know yet about for the like pathogens. I think they are important too. I don't know in the EAE context because probably you have to test. In the knockout mice, I have to remember because I don't know if we did. Yeah, these mice, they have fevers. Probably I don't remember from top of my head what's the FOXP3 populations. But they kind of delete the commensal ones, and they have colitis, right? Some point after like let's say after 3 or 4 months. If you have a how can I say a mouse room that have something that could induce like inflammation. In Colombia, we don't have that. We didn't see any kind of these things, but I know from other groups that they kind of develop like colitis later. So probably they are important too, but I don't know. In the I have probably to check. So one thing I'd like to do is kind of process mice maybe with CD. I think we have here right CD11 secret mice. So to distinguish this and see the different parts of this.
Oh, we also have a question from Vinicius.
Leandro, nice talk. When I look in the picture about the distribution of the vesicles, I'm so curious because in intestinal epithelial cells, we have a gradient from basolateral to the axial surface, and this is maintained by some proteins like the galactins. And I think you remember me a picture from a paper that some bacteria try to translocate into those cells, and galactins maintain those bacteria in the surface. So, you look at those proteins, what do you think that these vesicles could translocate to the basolateral surface?
Yeah, at that time I didn't have the chance or time to do that, to look probably I don't know if probably they overcome this, or I think actually they do go to the phagosomes. If I'm not wrong, they go to this. And the thing we don't know is how after that what happened, right? If it's releases like in the lamina propria of macrophage are phagocytosing these dead intestinal epithelial cells, would be different things. I don't know if it's just CDC42 because you saw that is like a 2 or 3% or 4% of J17 cells induction. Probably they could go not just inside the intestinal, but probably basolateral. They can, or maybe even the dendritic cells could acquire other antigens. But I don't know about what specific for your question about if is what kind of proteins are involved in this process. What we just demonstrated is not like protein dependent, and it's like dynamic in CDC42. We didn't investigate more deeply how inside the cells.
Okay, so we also have a question from Montilo, right?
Welcome to the party.
Thanks.
And I really am a little bit confused about the immunity of. I will stick to this type 3. So, and you advanced a little bit of what think about that. But I would like to ask first, antibodies are important, IgA is important, macrophages are important,
Yeah,
neutrophils are important, lymph nodes are important.
Yeah.
How you figure everything and what control ultimately the infection?
Yeah, so this is a very complex, especially the gut, right? You have a lot of different things. Yeah, everything, as you mentioned, is controlled in this situation. Like the antibiotics could affect the neutrophils. It's kind of difficult that's why it's very difficult to study this, right? Because we have a lot of pathways that could overcome or overlap in these conditions. How can I say? So basically, I forgot, sorry. Ask me again the question, your question. So that you think is not.
You can start with IgA. It's more easy.
With IgA, yeah. So basically, yeah, the IgA is induced in this context too.
Or if you don't have IgA, what else?
Yeah, if you don't.
You have
yeah, in this case.
Without IgA.
Yeah, we have IgA, yeah. So anyway, so I think it's very complex. So I think ILC3s are important. And that's why I think they are kind of brief, because they kind of communicate with different cell types, with T cells, with B cells. They can produce IL-17 and recruit neutrophils. They could produce IL-32 and affect the intestinal epithelial function. So that's why I think maybe I have to test, right, to know. But I think you're right. It's very complex, right? It's not just type 3 immunity or type 2 or type 1. We have a lot of cells that could work in this, especially the gut, right? So we have a lot of, like a second or first maybe immune system, right? So.
Okay, so there's a question at the chat too. So that's the question. Great talk. I think the experiment with mice and without lymph nodes is very interesting, but I'm a little shocked with the mortality rate. Did you notice any symptoms other than weight loss?
Yeah, I did. What we always analyze is weight, but we look at the feces too, right? We collect every other day the feces. I think was very higher burden in the feces, so we have like a very liquid feces in this. I didn't do like a necropsis to look like how probably is very inflammatory the intestines in this condition. But the main thing we observe is like the weight loss, and it's very quickly because I monitor the mice every day, but when they kind of die, they kind of die overnight. It's like probably because without lymph nodes, they kind of spread everywhere, like liver, they go to the spleen and the colon, and like spread a lot. So probably, I don't know, other symptoms for sure. We have the spread for other tissues, like that's why they probably die from sepsis or a very high infection.
Any more questions here in Rio?
Another one.
Oh, there's another one. Okay. This is in Portuguese. Professor Leandro, fiquei intrigada com a palestra quando percebi que o senhor citou o Citrobacter. Entendi que a ILC3 e outros atuam na defesa do organismo, mucosas, quando atacados por esse micro-organismo. Está correto?
Outras células provavelmente atuam na defesa.
Sebastiano,
Yes, the LC3s are important, the T cells too. But yeah, let's say as which will say, macrophage neutrophils kind of helps in the clearance, right? So we have other cell types like macrophage, neutrophils that helps to the clearance of this pathogen. So if you look like in the gut, I didn't put here, but I show that you have like increase of pro-inflammatory cytokines, you have like neutrophils, macrophages in the large intestine. So basically, the innate lymphoid cells looks like they are the first responders, but after the clearance of the pathogen needs like other cell, like effector cells, like from the innate immunity too.
Okay. I think we're done here, so I'd like to thank.
thank you. Thank you, Professor Leandro.
Thank you. All right, yes. Mas, pessoal, boa tarde a todo mundo.
Okay, thank you very much.
Thank you.
Next.